Hi, everyone. Before I get started, I want to thank the organizers. It's been a great conference so far, and I'm really looking forward to the rest of the conference. So thank you for having me here. My name is Vahab. I'm a biochemistry graduate student at Georgia Tech. I've got my email on here. You can feel free to reach out to me there, but also feel free to reach out throughout the rest of this conference as well. So today I want to talk about water's role throughout the evolution of life, and we're going to talk about it from the Hadean Earth all the way to modern biology. Okay, so first I want to take a step back and kind of take a bigger picture view of what's going on. I think it's really easy sometimes to overlook the things that are right in front of our eyes and kind of all around us. So if we take a look at the entire planet, we look at some life forms like animals like birds, fungi, bacteria. What are all of these things sharing with each other? What are the common features that they all have? Give you a moment to think about that. This is also something that you have as well that's shared with you, and all these other things. Everything that's alive needs this to survive, and you may already be thinking from the title of the talk, and it's already been talked about several times throughout this conference, but I'm referring to water. And we all know that water is important. I don't think anybody here is really going to dispute that water is important for life. But at the same time, I think that there is a lot that we need to learn about water and how it influenced life, particularly at its origin and the dynamics that are played with all these other processes that were going on. So we recognize that it's important, but I'm hoping to kind of share that maybe it's a little bit more important than we realize sometimes. So obviously water is everywhere in life. It's can be thought of as a metabolite and the most abundant metabolite in life at that. So if we have this pie chart here with some of the metabolites in life like glutamate, ATP, things that biology and metabolism use a lot of. If we put those on the same scale as water, we see that the water involvement in these processes just completely dwarfs the involvement of some of these other molecules, really emphasizing how actively water is used in biology. Water has been around since the early Hadean. Pretty much as soon as the Earth was cool enough to have stable liquid water, it did. And at that time, given the environmental factors, it wasn't boiling at 100 degrees Celsius. I think water was like liquid up in like 200 degrees Celsius. So not quite the same conditions, but still liquid. So has been around for a very long time, and it definitely plays an important part in how we think about our search for life. We sent a rover to Mars to a particular location that we think had water running at some point. There's been a lot of interest in the icy moons. I think Europa was mentioned later. I think this is of this picture here is a representation of what it might be like on Enceladus, where we have these plumes. And Venus was mentioned yesterday as well, and I think there was some reference to water being present there as well in the clouds or earlier on in its history. So water is kind of at the forefront of how we think about exploration in space, especially when we look for life, clearly outlining how important it is. And yeah, without water, we couldn't exist. But at the same time, as important as water is, it's also dangerous. And I think it's important to talk about how dangerous water is. And dihydrogen monoxide is another name for water. It's colorless and odorless. It's accidental inhalation can be fatal. So referring to drowning here. This is a little bit of a joke slide, but water can be dangerous, and we can definitely frame it this way, but that's not quite what I mean when I say that water is dangerous. I'm more talking about how dangerous water was when life was trying to get started. And so at some point in Earth's history, there would have had to be a transition from the building blocks and the organic inventory present to polymers, and then those polymers would have eventually, whether it was RNA, protein, something like that, would have then transitioned to life. But at some point, we would have had to form polymers. Now, the problem is that in water hydrolyzes biopolymers, and that's because they're thermodynamically unfavorable. If you look at this free energy coordinate here, you can see that the monomeric forms are more stable than the polymeric form in water, which means that if we form the polymer, they have a tendency to be hydrolyzed by water and be broken down. We can see this also illustrated with some of the the representatives of biopolymers in life. So all of life has proteins, it has nucleic acids like RNA and DNA, and it has polysaccharides. These polymers are essential to all of life as we know it. And every single one of these, the polymers can be broken down into their monomers in a high water activity environment. And those monomers can also be driven towards polymerization in a lower water activity environment. So it kind of outlines this dynamic relationship between water activity and these polymers. And so if we imagine these on the early Earth in an environment with water, it would have been a struggle to survive. So how did we end up with biopolymers in life today? How did they survive this process? Well, I won't be answering that question explicitly today, but we are going to look at modern biopolymers as an example. So we can look at biopolymers today to get some answers. And one interesting thing about all modern biopolymers is that they can assemble. So this assembly can confer an advantage to the polymer, allowing it to survive longer. We call this recalcitrance. So that's just the ability of a molecule to persist for a long time. This is an electron scanning image of cellulose in a water inclusion that was dated to 250 million years ago. So for 250 million years, this assembly of glucose or of sugars has been stable enough to maintain its assembly, which really speaks to how powerful assembly can be in protecting these polymers. Now, what does that look like for this free energy diagram? Well, once it's assembled, it becomes much more stable than the polymeric form. Maybe not as stable as the monomer. It can still tend to lean towards hydrolysis, but much more stable than as a polymer. That is a non-assembled polymer, I should say. And this essentially imposes a selective pressure during the process of chemical evolution when these monomers would have turned into polymers. Again, this is an image of modern biopolymers. They all have assemblies. We have proteins here taking on various assemblies, sugars taking on assemblies, bases. This is, I think, DNA here taking on an assembly. So all of this assembly confers that advantage in that it can survive hydrolysis and survive in water. So how does this happen? Well, this is something that we've probably all observed. In the morning, we have water on a surface, and you come back later in the day and that water's gone. So what's going on here? This is something that we call wet-dry cycling, and it comes down to the fact that the Earth rotates. The Earth has always been rotating, and this results in a diurnal cycle or a day-night cycle. At nighttime, when it's cooler, you'll get that condensation. So in the morning, you see that water on that railing, and in the afternoon, or as it's warmed up, it dries off and the water is removed. So this creates water cycling, water activity cycling, and we call that wet-dry cycling. And this is an experimental platform that we can use to understand what was going on at this stage of the origins of life. But I'll talk a little bit more about that later. But first, I want to talk a little bit more about what's going on the early Earth. So in addition to the Earth rotating, we also know that there would have been land-water interfaces present on the early Earth all the way back to the early Hadean, going back to like 4.4 billion years ago, also pretty soon after the Earth formed. So these have been around for most of Earth's history, and they're important because they allow for a thermodynamically favorable environment for these biopolymers to form, to take those monomers and convert them into the polymers. So yeah, the Earth is rotating. We have land-water interfaces, and well, what are we reacting with? What's on the land or what's in the water? So we also know that the early Earth would have had organics raining down on the surface. There's different models on how this would happen. One situation is after an impact, the atmosphere would be reduced, and then that would allow for the formation of these organics to rain down. This image here is kind of a similar concept, but this is Titan, which also has a reductive atmosphere, and similar processes may be happening there. But essentially, on the early Earth, these organic molecules would have been raining down onto the surfaces. And somehow these organic molecules and this chemical inventory would have turned into life. And again, so that those needed to survive turned into biopolymers, which were able to survive in that water environment. So what happens if we, how can we do this experimentally? So I've talked a little bit about the balance between high water activity and low water activity and how it drives the formation of monomers or polymers, respectively. And this little blue chart here, you can think of that as the day-night cycle going through the history of Earth. So throughout all of Earth's history, this has been happening. If there's been water, if there's been land, if there's been a chemical inventory, we would be going through wet-dry cycling. So our hypothesis is that this water activity drives chemical evolution. This cycling imposes a selective pressure where in the wet stage and high water activity environments, the molecules that can survive better and resist hydrolysis persist and continue on and survive. Now that selection the selective pressure can be imposed on a few different scales. One is at the scale of solubility, so what's able to even dissolve into water and what's not is going to be the first level of selection. We also need to consider the ability of the molecules that are going into solution to be able to participate in the condensation chemistry needed to drive polymerization. After that, we need to consider resistance to hydrolysis. So there's different types of bonds that could form, which ones are going to be able to resist hydrolysis better than others. That's a consideration that's to be made, and there's going to be selection at that scale. And then we get to selection of which molecules can assemble, like the cellulose that we saw surviving for 250 million years, as opposed to another molecule, which we probably won't see because it degraded into monomers. So this selection occurs at a few different scales, and we can experimentally do this by wet-dry cycling a chemical mixture and seeing what happens. So just to illustrate this model a little bit better, we have our organic inventory, our molecules on the prebiotic Earth. We have our chemicals, our organics being delivered, and then we get these selective pressures on solubility. So these are molecules that survived through their ability to be soluble, continued on to be selected upon based on their rates of condensation and ability to form oligomers, dimers, trimers, so on and so forth. And then we go on to their ability to resist hydrolysis. So the things that aren't being broken down are going to survive. And then we get to assembly. There might be some steps missing in between here. And there are many other steps that came after before we got to what we consider life today. But this is a general outline of what that may have looked like. I have a QR code to the preprint of this paper where we outline this model a bit more if anybody's interested in that. But some experiments that we've done in the lab, we've conducted wet-dry cycling experiments for 15 cycles and done some broad level analyses to try to understand the behavior of the system. And we see that the system continues to evolve. Here at the bottom, we have, in the gray, we have our cycle 0. And then as we move up, we are increasing in our cycle number. We can see the changes in these different types of peaks. This is an HPLC chromatogram. We've used a few other types of analyses too to just broadly look at the behavior of the system, but we see this continuous change across these cycles, which for us is promising, and that tells us that the selective pressure is driving chemical evolution.
You have 1 minute.
Okay, thank you. So this slowly ratchets the system towards longer molecules, assemblies, things that persist. Now, next we are trying to do more cycles. So 15 cycles is nowhere near what has happened through the course of the origins of life. And so we're creating a robot. This is me inside of an anoxic chamber, setting up a robot that we're going to be using to do this for much longer timescales. And I want to wrap up with some things that I hope you can take away from this. I hope you can just keep water in mind more. I want to remind you that these land-water interfaces have been present for most of Earth's history. The organic materials have been present on Earth's surface for most of Earth's history as well. And the cycling of this water activity at these interfaces imposes a selective pressure upon these organic libraries, which would drive chemical evolution. And taken together, we can create a cohesive model of the origins of life, which puts water at the forefront of what's going on. With that, I'd like to acknowledge the grants that supported this work, my advisor, Lauren Williams, some of our collaborators, as well as the whole Williams lab. And I'll leave you with this quote from Lucy Larcom: "A drop of water, if it could write out its own history, would explain the universe to us." Thank you for listening. This is the QR code to that paper that I was referencing, if anybody would like to take a look.
Oh, there is the
Yes.
the QR code. And